Good afternoon, or I guess we're still morning. My body's in afternoon time, but I hope everyone's doing well. So, a pleasure to tell you a little bit about Arbor. We are a next-generation gene editing company. The company was co-founded. Maybe this will advance. Oh, it's like the old school remotes. It just needs a wire. So the company was co-founded by Fong Zhang, David Walt, Fong being the CRISPR pioneer and David Walt being an expert in high throughput automation and robotics, co-founded a company we all know called Illumina. And the two got together and they actually founded the company on an idea as opposed to being based on IP. And that idea being that Cas9 was the first nuclease found, but there's likely to be many other differentiated editing approaches out there that evolution has developed and designed over millions of years. So flash forward several years, we now have a very robust discovery platform that has allowed us to build what we feel is the most extensive toolbox of proprietary editing approaches that are all wholly owned. We've really focused in on thinking about the disease we're targeting first and then bringing to bear the right tool. Another important piece for us is, while we have a really interesting platform, we have to very quickly translate these into new novel therapies. And so we have a goal of 3 IND or CTA filings over the next 3 years. Our focus is in diseases in the CNS and liver. And our goal is to be the first non-Cas9 nuclease tested in vivo in humans. Now, the team and investors are obviously very important. We've got a very strong cohort of investors, have raised over $300 million to date, and we have a seasoned team that has achieved over 20 INDs in cell therapy, gene therapy, and gene editing. So we know how to translate these novel discoveries. The platform itself, just to give you a little bit of view on how we discover things, is there's a whole lot of data out there. We have a lot of privately sequenced data. There's a fair amount of public data, but it's trying to make sense of that data. We've got some very talented computational folks that do things that are way outside my level of understanding as a geneticist on how they do it, but we're able to really quickly identify large numbers of potential interesting editing systems. We have built a large high throughput screen in bacteria that allows us to quickly characterize a system. If we're looking for a nuclease, we can very quickly identify the PAM, the size of the system, the cut type, and what the off-target profile looks like. Really quickly honing in on those systems that may be interesting to us. Now, we can identify hundreds of thousands of systems, but we're not here to collect baseball cards. We're here to develop therapies. And so one of the pieces that's interesting is once you identify one of these systems, that's the easy part in many ways, because then these don't tend to work well in human cells. Because we're focusing on non-commensals, things found in deep sea vents and other areas where humans haven't been, these tend to work very poorly in human cells. And this is where the experience of some of our folks in the company, having worked in large companies where we know structure-based approaches work. So we can generate structures of these systems. We can also use AI systems like AlphaFold from Google to very rapidly identify structures, generate the SAR, structure-activity relationship, so we can very quickly know what residues we need to modify to get on-target activity up, as well as to get the off-target activity optimized. That allows us within a couple months to have in this case, a nuclease that's extremely functional, at or better than Cas9. Now, focus is obviously important for us. As we think about all the things you'd want to do to treat genetic disease today, having this robust suite or set of chassis of nucleases allows you to think about, okay, where else do we want to deploy this? So it's 4 areas we're focused in on. One is on knockdowns, where you can really downregulate a gene expression. That takes care of a set of genetic diseases. Second is precision editing, or coupling the nuclease to a reverse transcriptase. That gives you the ability to rewrite sections of DNA, and that takes another sort of set of genetic diseases. Then we've got diseases where you have large numbers of repeats, where you need to excise something. In this case, examples, something like Huntington's, where you need to excise a large number of repeats. And again, having the ability to precisely excise that is important. So that's a third component. And then fourthly, thinking about large insertions, the ability to drop in a large chunk of DNA into the endogenous genetic locus. We're not looking at landing pad insertions. We're very focused on going into the endogenous locus. So those are the areas of focus. Now, why that matters is that it allows us the flexibility to actually pick diseases that we like and then bring the right tools to bear. And so that gives us a lot more flexibility in thinking about what's the best diseases for us as a company to focus in on versus those we may think about partnering. The other piece, I think it's beginning to be borne out in the literature, and we've seen this a lot, is by screening a lot of different nucleases against a particular target, at this point, we cannot predict which nuclease is going to be the best. And that's because there's a lot going on at the DNA level on a particular locus that I think we're just beginning to understand a lot of those components. But it's clear having the ability to screen hundreds of guides against many, many nucleases allows you to pick the best approach for that particular disease. Now, where are we focused? As we think about how do you pick your strategy, this is really important. We wanted to start somewhere where there's clear delivery. So liver is a great place to start. We know we can deliver via LNPs. We also wanted a disease where there are opportunities that are clear clinical, there's clear regulatory rationale and approaches, and finally somewhere where we could be the first editing approach. So we selected primary hyperoxaluria as our lead. That allows us to not only build the expertise in-house, but also be able to take that expertise and leverage it to move into other areas. So moving into other liver diseases where we can target via, say, an RT or precision editing, and also going deep into the CNS. So as we think about the pipeline, again, with that focus of 3 filings in the next 3 years, we really want to start with effectively a gateway indication. One indication for the technology at hand, go deep on it, understand the technology, all the parameters of the technology, that then allows you to be much faster and quicker and less expensive for the subsequent diseases that may use that technology. You can see here we have a mix of liver and CNS indications. The other note is we are not doing any ex vivo cell therapies. We're partnering in that space as we have enough to do in the in vivo space. So we do not work in the ex vivo space at this time. Delivery is another really important component here. And I think there's a lot of opportunities from the delivery side that folks are working on externally. Our view is we are not a delivery company. We are not going to build our own delivery platform, and we're not going to we will build internal delivery expertise, but we will make sure we partner to access the best delivery approaches that are out there. So we do have a partnership with Acuitas on the LNP side, and we've got several things in the works on the AAV side to make sure, as we think about outside of the liver and in the CNS where we want to target, AAV gives us the near-term opportunity to target specific diseases. And so now we've got the strategy in place. Let's look at how we think about targeting a disease. So primary hyperoxaluria quickly is really there's a mutation with little red X's right above AGT, which prevents the formation of glycine. So what happens, if you remember way back in the biochemistry and all the cycles you probably forgot about, is that it shunts everything to oxalate. Oxalate is insoluble, which it forms crystals, which destroys the kidneys, and these patients end up on dialysis and end-stage renal disease in their early 20s. So there's an opportunity to intervene at the HAO1 gene and create basically a dam there, which builds up glycolate. Glycolate is water soluble. So an important piece of the puzzle is how do we think about creating a novel drug? And this is something that this approach can apply to any disease that we look at. But really, we start with a significant amount of in silico target design work. We've got a really talented group of folks that leverage all sorts of novel algorithms they built to quickly identify the right approach, screen out a lot of approaches that may not work so that we can very quickly focus in on a small number of things we need to screen. We'll take those, in this case, some particular guides and nucleases, do an in vitro screen in HEK cells where we can screen many hundreds of things, take the most interesting pieces of that and move to our primary cell, in this case, primary hepatocytes, validate that they do indeed work and translate well from an editing perspective in the hepatocytes, and then we will then do an off-target analysis. And that's an important piece of the puzzle. The other side of it is really thinking about the potency of the guides, because guides potency differs depending on the guide you're working on. So this process is something we can take across all the different indications we target. So with this work done, we can move into animals. And we think about the animal work, what we have here on, let's see, the left-hand side, yes, your left-hand side, this is a really important time course experiment done in mice that looks at three things. The blue is looking at editing over time. The orange is looking at the Go protein, so the protein of the gene that's edited, when does that go down over time? And then thirdly, what's the impact on your biomarker? And you can see in here, editing begins in the first, call it 4 to 6 hours, you start to see it, and it really peaks at about 2 days. This is delivery with, again, an LNP carrying a messenger RNA and the guide RNA. You also see the protein levels begin to drop, and they really drop significantly once you've eliminated about 75% of the protein. And you see that biomarker really shoots up significantly at that point. In addition, we can look at dose dependency. We see nice dose dependency on the indels, and we see nice dose dependency, again, on that serum glycolate biomarker. So it really helps us understand the kinetics of what we're looking at from a putative drug perspective. There's also a mouse model that one can utilize. So in this case, again, we can looking at dose dependency on both the indel side of things, but also on the right-hand side in the upper right. What's the impact on the protein? You can see in this particular experiment, about 90% of the protein was knocked down in the low dose and all of it in the high dose. Then we know what's the impact on the urinary oxalate, which is the biomarker in this particular mouse model. You can see that that biomarker drops very quickly and significantly. Now, an important point here is you actually don't see distinction between the dose levels. And if we hadn't have done the prior experiment, we'd be scratching our heads. But we know, based on the kinetics of what we saw in the mice, what we should have seen. And indeed, we did see that. So it recapitulates the biology, gives us confidence to move forward. We've done a significant amount of NHP studies at this point. We've put in place the appropriate supply chain components, so we have our CMC all set, and finally, we've had multiple regulatory discussions. So we feel we have a clear path to the clinic for this program with a targeted filing next year. I have 2 minutes, so I'm trying to push, but ALS is an area we're interested in. There's a lot of targets in ALS. And just an example of how we think about, in this case, an excision disease. One thing we want to do is screen, again, across a lot of nucleases, a lot of different opportunities to make sure we have the right scientific approach for moving forward. So we do that in things like HEC cells. Pull the most interesting approaches over into patient-derived iPS neurons, and again, test, do we see high levels of excision? You can see in that middle panel, indeed we do, almost 90 to 100% precise excision. So reminder, you're trying to remove a 2 to 3 kb repeat out of these each cell. The other piece on the far right is we also want to make sure you maintain the wild type level of transcript. Again, you don't want to impact the wild type functionality of this transcript. You want to get rid of the repeat transcripts, and indeed we're able to do that. So again, just showing the power of the technology and the ability to move the different technologies across indications. So in summary, we are a next generation editing company. We've got 4 different editing technologies that we enable by a gateway program approach, a very experienced team, and we're targeting 3 IND and CTA submissions in the next 3 years. So thank you, and there's 49 seconds.